Good morning, everyone. Welcome to today's adult education session. I am just so thankful for Denise Spooner reaching out to us about an incredible speaker today. It seems like a lot of you know him already, which is great, and may have heard him present. And I am very interested in what he's going to talk about today. So this is Dr. Larry Grill, who is the Dean of Research at the Keck Graduate Institute, where he teaches courses on virology, pharmaceutical development, and biotech-based therapeutics. He teaches undergraduate courses for the Keck Science Department at our colleges here in Claremont. And with his research lab at KGI and with collaborators in Botswana, he is working on creating low-cost vaccines for developing countries. Their current emphasis is on COVID-19 vaccines. He received his Ph.D. at the University of California, Riverside. He has over 30 peer-reviewed publications and is an inventor on more than 30 US and European patents. And so he is here to talk to us today about disease control and COVID-19. So thank you for being with us, Dr. Grill. The floor is yours.
Okay, thank you. I assume I can share screens. Okay, let's go there. All right, now am I assuming you can see that screen? Okay, good. Okay, so I think it's really important right now to kind of let everyone know what these vaccines are about for this COVID-19, because it has been a rush to get these vaccines out. We do have a pandemic, we do need to do something. But I think just a little bit of knowledge about vaccines and what they're all about is really worthwhile at this time. So I'm going to kind of tell you a little bit about the different vaccines, the way I see them, and how I'll describe some of the ones that we've already heard of, Pfizer, Moderna, and AstraZeneca, and describe those and how they're different and how they're the same. And also just to give you a little more comfort on what they're about. I'm very anxious to get my vaccine shot, and this is where age is kind of to my benefit, I think. So let me go ahead and get started here. So I like to always introduce vaccines in general because vaccines have been around for, well, since 1796 is when they first started developing it. And the one that we know about is the one that was smallpox. And the way it happened is there was a knowledge that people who milked cows, if the cow had a disease called cowpox, they would get cow pustules on their hands. And it was a disease that was very short-lived. But the good part is it ended up giving them resistance to smallpox. Now, smallpox has been one of the worst pandemics we've ever had. It literally killed a third of all the people in Europe. So that was a pandemic that was really disastrous. So you'd lose almost the whole town. When it came over to the Americas, we lost 90% of the people to smallpox. And so something needed to be done. And so Edward Jenner, who recognized what was going on with this cowpox, said, hey, let's take and inoculate this little boy, a 9-year-old boy, with the cowpox. And we knew that cowpox only caused these cow pustules on your skin, and then it'd go away in a couple of weeks. Well, it turns out what he did later was something the FDA cannot do now, but he inoculated the boy with smallpox, which would be a risking death since it was killing 1 in 3 people. But it turns out the boy was resistant to smallpox. And so he started developing this and basically giving it to everyone else in the towns nearby. And within a matter of time, the huge amount of population were vaccinated with this cowpox, and they were resistant to smallpox. And right now, the only human disease that we have ever eliminated on this planet is smallpox. So it does not exist on this planet anymore, and it's primarily because of the vaccine. The fun part about this story is the word vaccine comes from vacca, which is, and that's from the cowpox. So vacca is Latin for cow or Spanish. And that's because they were using cow pus to vaccinate people. So that's where the word came from. I always think that's a fun part of the story to know where vaccines originated. Okay, so now to kind of describe what the viruses are doing when they get in your body, so you'll understand what the vaccines do, I'm going to introduce a cell, and the cell is going to be a cell that you normally would see in your body. You have over 50 trillion cells in your body. And so this is kind of the standard cell. What they all have in common is they all have a nucleus. So this is the center part and has your DNA. And you've all heard of DNA, and it's always in the nucleus. But in order to get that information out, because we need to make proteins, which are going to be enzymes and your hemoglobin and all these other things are proteins, we need to make them. And the instructions are on this DNA, and it's sent out into the manufacturing part of the cell, which is using something called a messenger RNA, which is just what it sounds like, a messenger, to make a protein. And these proteins are what we're going to be using for our bodily functions. So that's a typical cell, and that's what it normally is doing. Busy, DNA to messenger RNA to proteins, cycle back and forth. But then when the virus comes in, the whole world gets changed. So what happens is the virus says, I don't want you competing with me. So basically it's going to shut off this messenger RNA coming from the nucleus. It will shut off making that protein because the virus wants to be in control. So here's our infecting virus. It's making its own messenger RNAs, and these messenger RNAs are making its own proteins, and these proteins are being used to make more viruses to go out and infect other cells. So it's slowly taking over your body by its ability to just shut down the cells and use the cells for its own function. Now, we aren't without some ability to fight off these viruses. We have several different ways of doing it, but I want to introduce to you what this cell does the first thing when it starts being infected. It realizes it's being infected. It realizes it's making proteins that are not its own proteins. So it basically says, okay, I need to show my white blood cells what's going on here. So what it does is it takes some of these proteins that are virus proteins, puts them up on the surface and says, hey, come on, guys, take a look at this. Come on, white blood cells, take a look at these different proteins. They're wrong. They're not you. They're foreign. I'm in trouble. And so that's what it's doing to kind of send the signals out to your white blood cells. Now, I need to also talk about a very important part of your white blood cells. Basically, there's something that we call antibodies. And maybe many of you have heard of antibodies, and we do know that there's some antibodies from serum from people who have recovered that's being used to help people who are infected. There's also factory-made antibodies from Regeneron and Eli Lilly that are being used for people who are infected. But these antibodies are very important because what they have the ability to do is at the tip of these antibodies, they are able to bind tightly to the COVID-19 virus spike protein that's on the surface. And when it binds to it, it won't let go. So we call it kind of a lock and key. It will only bind that virus. It will not bind anything else. And so these are going to be very important in fighting off the disease. So let me show you what the main three white blood cells that I want to talk about. There's several others going on, but I just want to talk about three. We have what I call the first responders, and they're going to be your macrophages. And these macrophages are going to be gobbling up anything that's foreign as fast as possible. And so most of the viruses that you get, that you don't even get sick from, it's because the macrophages, your first responders, get rid of it right away. Unfortunately, viruses and a lot of bacteria are pretty sly, and they know how to evade the macrophages. And the SARS-CoV-2 virus, which was the COVID-19 virus, that SARS-CoV-2 is very effective at evading these macrophages. And that's problematic. So what we do have is we have a backup system, and I'm going to give them the names B cell white blood cells, B cell lymphocytes, T cells, and the B comes from where they originate, the bone marrow. The T cell comes from the thymus. So these white blood cells, that's just their place of origin. But they're our backup team. The bad part is they have to amplify and make a lot of them because they're the ones that have these very specific antibodies that I just showed you back here. So these are going to be made by the T cells and by the B cells. So here we have the B cells and the T cells. So they're going to be our backup. The bad part is they take about 4 days to get to build up enough of them and to start making these antibodies that are going to specifically bind to the virus. And so they're very important to do it, but now we're going to be sick for 4 days because our first responders couldn't handle the disease. So the macrophages, again, the first ones, like I say, unfortunately, some of them evade these first responders. So let's talk about the B cells and the T cells specifically. So the B cells are going to be the backups coming in here. So here I have a picture of a B cell, and it is actually throwing out antibodies into the bloodstream. So these are naked antibodies that are out there being made by this B cell and throwing them out there. What this does, this is super. So now you have these soluble antibodies that are going to go bind the SARS-CoV-2. And what that does, that gives your macrophage, the one who missed out at the beginning, it now, so here we have the virus and it has these antibodies binding to it. Now your macrophage says, oh, now I can help again. So it now has additional signals. And so it starts gobbling up all these soluble viruses that the antibodies are bound to. So it's very important because we need to get rid of these viruses that are soluble in your blood. And that's what B cells do. But we also have to worry about something else. So while you do have these soluble SARS-CoV-2 viruses running around in your bloodstream, you still have a lot of infected cells, and these infected cells keep making more. So we have to do another battle, and that's where your T cells are going to come in. So your T cells are going to be putting parts of these virus on its surface to train T cells to go after them. So all these cells that are infected are going to be displaying these proteins, and we call them MHC presenters, but basically don't worry about the name of it. But what they're saying is these cytotoxic T cells say, I see that this cell is infected, I'm going to kill that cell. And you want to kill the cells that have the virus in it because they're up to no good. They're not going to recover. You got to get rid of them. So the important part about these two sets of cells is we need them both to really effectively fight a virus. You need to get rid of the soluble viruses. You need to get rid of the viruses that are infected in cells. And so you need to get rid of those cells. So that's our double system that we have to go after. Very important to go after those. So again, just summary, we need them both. And I'm going to show you how using the antibodies that we have and the B cells and T cells, we can effectively fight the SARS-CoV-2. So that's where we're going to start talking about using vaccines. Okay, so just what I wanted to also let you know, when you effectively fight the disease with your B cells and your T cells, life is good because you now have memory cells. So they say, okay, we've eliminated that and we're going to keep basically a set of soldiers behind, and we call them memory cells, memory T cells, memory B cells, and they're going to be hanging out waiting for that virus to come back again. And if it comes back again, it's going to kick its butt just within a day. It's gone. However, what I always want to point out is sometimes the B and T cells don't win the battle. This is the case for 315,000, over 315,000 people in the US who have lost their lives to the SARS-CoV virus. So this is what we don't want. We want to have our B cells, T cells ready to fight. We want to train them ahead of time so that we don't lose. And that's the whole goal of the vaccines. So we're going to use vaccines to train our cells. And what's important here is we have some new vaccines out that are just mind-blowing and they're just breakthrough. This is going to be so important for us because, as many of you know, it generally takes 10 to 15 years for a new vaccine to come out. And that's because we just have a standard old way of doing it. Now, remember that we do come out with new flu vaccines every year. That's because we're using the same technology year after year after year. and they're good for that particular strain, but then the next strain comes out the following year and it's not good on that. So we have to retrain our white blood cells again. We have now the capabilities of actually doing a better job of training our cells with B and T. And I'll tell you how this is really going to work. First, I want to describe the different vaccines that are out there, because again, we have, you've heard Moderna, you've heard Pfizer, you've heard AstraZeneca. I'm going to tell you what these vaccines are and how they work and how they're different. So I like to basically, this is my own designation of the types of vaccines that I see. I put them in classes. The one here called inactivated viruses, that's going to be the typical flu vaccine. That's where they basically grow them up in eggs, in egg embryos or in cells, and then they extract them, purify them, and kill them with formalin. And then those are just used as your vaccine. Those have been around for 60 years now, just making them that way over and over again. Nothing new coming up, but it's been tried and true. And everyone says, let's just keep doing the same old, but we don't have time to do that right now. So we have some new methods, and I'll show you them. But first I want to there's one that you may have all had at one time in your life, and that's a live attenuated virus. That's where the virus has just been mutated a whole bunch and cannot fight a good battle in your body. And that would be a good candidate. That would be the oral polio vaccine. Even though we don't have the oral polio vaccine in the US anymore, in the 50s, we did have some oral polio vaccines out there. And you just took it on a sugar cube. It went in your stomach and system, and basically it grew, but it was so weakened, your body could easily put it down. This is a little more dangerous one, because if you are immunocompromised, your immune system may not be good enough to fight this. So we're not using this one for the COVID-19 disease, but it has been out there, so I wanted to just introduce it. The one that I'm working on, and the one that is also out there by several different companies, called a virus subunit. And this is going to be a virus piece. So not the full virus, so it can't cause an infection. And the problem with some of these, like this inactivated virus, since it never gets into your cell, it's not going to stimulate your T cell response. And some of these virus subunits don't trigger your T cell response either. And so they're not going to do as well against viruses. The new class that we're looking at, the live recombinant, the messenger RNA and DNA, these all stimulate T and B cell production. And again, I emphasize, we want both T cell and B cell response to effectively fight this virus. So just a little more detail. Again, the inactivated virus, we grow them up, purify them, kill them, and inoculate them into your arm. Then the attenuated viruses, they basically grow them up and many cycles, mutate them in many various ways. They can do that, purify it, and it's a mutated DNA, and that one's used sometimes. In fact, there's something called flu mist, which is a vaccine that's sprayed into your nose. That's an attenuated virus, so it's made similar to this system. Here's the subunit vaccine, and as I mentioned, subunit is exactly what it says. It's a subunit of a virus, subunit of a bacteria. So it's just gonna be a piece of it, just enough to kind of train your white blood cells, but it does not have the ability to cause a disease. These are very safe. And the one that is most knowledgeable to younger teenagers is going to be the one for human papillomavirus, which is a virus that is responsible for cervical cancer. So this Gardasil, as we know it, is basically going to be just the coat protein of the virus, no nucleic acid. It can't cause a disease. They make it in a yeast, so they use common bred yeast to make this protein. They let it assemble into this capsid. It looks like a virus. It trains your white blood cells. And now you're being trained against the disease should the real one actually come. The one that's AstraZeneca is called a live recombinant. And that's because what they're doing on this, they're taking a symptomless virus. And when I say symptomless, this is a virus that knows how to get into your cells, knows how to replicate, but it doesn't know how to cause disease. In fact, it's been altered so it has no ability to cause a disease. What they do with this symptomless virus is they take the genetic information for the spike protein from the COVID-19 virus. And that is now going to be made and put on the surface. So now we have a symptomless virus that I say has a costume to look like the SARS-CoV-2. It can't cause a disease. It can't, so we're not going to have people dying from it. It's symptomless, but it is going to train your white blood cells. So I have a picture of how that has happened. So basically, with AstraZeneca's vaccine, they're using a chimpanzee, and it's called an adenovirus. It doesn't cause disease, but they've taken the spike protein from the SARS-CoV-2, and they put that in the genetic information. And so now they're growing it up in large scale. They now have a symptomless virus, but it's decorated to look like the SARS-CoV-2 virus. And so it's going to train, it's going to go in, it's going to infect, it's going to go in your cells, but it can't cause a disease, but it is going to train your white blood cells in the way that I kind of showed you that happens. So that's a really important vaccine out. It's not quite out yet, but it should be out probably within the next, well, by the end of this year, I'm quite sure. The ones that are out right now, both the Moderna, which just got approved, and also the Pfizer, which got approved last week, these are called messenger RNA vaccines. And this is a term that I introduced earlier. It's a normal messenger RNA that really does cellular function. What they do is this messenger RNA is coding for the protein, which is the spike protein found on the surface of the SARS-CoV-2 virus. And they introduce it into cells by putting it in a liposome. The liposome gets into the cell, it makes this messenger RNA, and this messenger RNA has no function in the cell. And the cell realizes this is not me, this is a foreign protein. So it does just what it would do if it were infected. It's going to display it on the surface, and it's also going to throw soluble pieces out in the cytoplasm. And this is the mechanism to train your T and B cells. So this virus, this cell, even though it's not infected, it thinks it's infected. So it's responding as if it were infected, but it's just making the spike protein, nothing else. But it's foreign, and it doesn't like foreign material. And so it's actually just warning your white blood cells saying, hey, come take a look. We have something going on here that's not us. This is foreign. And that's what the messenger RNA is doing. Now, the messenger RNA is common in both Pfizer and in Moderna's. They differ in slightly different ways. So I'm going to show you a picture of how these differ. So here's the Moderna. That's the one that just got approved. And these are under emergency use authorization approvals. So basically what it is a synthetically made messenger RNA, and it's made using nucleotide subunits that are going to not be degraded as rapidly as your messenger RNA. And this is really important because we need it to stay around long enough to make these proteins. So it's stable for a certain period of time. It may be stable only for a day or two, which is why you want to have the booster vaccine 2 weeks later, is because you really want to train these white blood cells, T blood cells, and B cells. So basically here we have the lipid covered messenger RNA, synthetically made messenger RNA. It goes in, it makes the spike protein. The spike protein is being sent out in soluble, and it's also being displayed on the surface. So out here is your cadre of white blood cells that are being trained about this spike protein. Now this cell is not infected. It's just making that spike protein, which the cell knows is foreign, and it's training your white blood cells saying, hey, we got a problem here. The Pfizer and also GlaxoSmithKline, they're about 3 or 4 months behind Pfizer. They have a self-amplifying messenger RNA. So basically, it's not synthetically made in the lab. They're actually just taking in an opposite copy along with something that's going to replicate it. So it's a single messenger RNA with a replicating enzyme. It's going to go in and it's going to make easily 100 copies of that. And that's what's going to make it last longer. Because again, these molecules are rapidly turned over. And so we need them to last long enough. They're not going to be permanent. They're going to be very transient, but they're both going to make the same messenger RNAs to make the spike protein and display parts of it on the surface of the cell. In this way, we're training our B and T cells. The other class that are way behind on the other ones is DNA vaccine. And basically, this is going to be the DNA. In the first slide I showed you, the DNA makes the messenger RNA. So this DNA has the genetic material to make the messenger RNA, but it's a longer route. And when I say a longer route, that means that this DNA vaccine has to go in, get into the nucleus. The nucleus is going to read it and then make the messenger RNA, which is going to make the proteins, which are going to be displayed on the surface. A little bit longer route. This one's been in development a lot longer than the messenger RNAs. It's been easily over 20 to 25 years that the DNA vaccine's been in development. It's being made for animals. There's 3 different DNA vaccines for animals, but we still don't have a human one approved. And I think the fear is, even though data doesn't seem to suggest it, the fear is, what if this DNA integrates into your own DNA? And so the FDA is really putting them under very stringent testing. So they still are not out. This messenger RNA has no ability to integrate into your system. We know it's rapidly degraded. The problem is we need it to last long enough. And so the booster and the way they've done it, it will last long enough to make to train your B cells and T cells. So these are super vaccines. Now, the messenger RNA vaccines have been in development for a little over 10 years. And they were in late phase and beginning phase of clinical trials for the Zika disease. And again, that was in the slow boat 10 to 15 years. But that when we suddenly had this emergency use authorization where we needed something right away to fight this pandemic, we have to stop these deaths. So they switched over, and basically in March, they started making the spike protein from the SARS-CoV-2 instead of the Zika vaccine. So they did a fast switch over, but they'd already done animal tests. They'd already done lots of proof of concept. They'd already been in humans. So a lot of this testing was done, but we just hadn't done it for this new virus. Now I want to kind of move over to the subunit vaccine that I'm working on. And the reason why I want to do that is because we are going to have enough vaccines by the end of next year for most of the United States and most of Europe. But remember, there's 8 billion people in the world. We're not going to have enough vaccines for them. There's still going to be lots of deaths. And so my world is making low-cost vaccines for the developing countries. I've been working on this for 10 years, and I'll show you where we've actually had some success on it. So what I use here, and this is kind of a fun story, I think. So we use a plant virus. Now, plant viruses are unique in they can only infect plants. And I, as a virologist walking around, I actually see trees that have virus infections. I actually see flowers that have virus infections. When I go to the farmer's market, I see lettuce that has lettuce mosaic. I see papaya that has papaya ring spot. So I see these viruses, but no one cares about plant viruses because they can't infect humans. They don't have the capability whatsoever. They don't know how to deal with anything inside our body. They do effectively cause disease with tobacco plants and other plants. So here's a mosaic symptom that we're seeing there, but that's what they do. But the good thing is these look like a virus. So that means since they look like a virus, you can actually use these to train your immune system if you change the outside surface, and let's decorate it like that. So let me show you what I mean on that. So let's go back and take a look at the SARS-CoV-2 virus, the virus that causes COVID-19. So here's the spike protein on the surface. It's easy to get that genetic information. And actually what we do and what we've done in the lab, and we actually started in April. So we were a little bit slow in starting here, but we said, hey, we got to switch over and start making this vaccine for the developing countries. And so basically we took that genetic information for the spike protein, put it inside the chromosome of the plant virus. So this plant virus, again, still can't cause infection of humans, doesn't have in any capacity whatsoever. So here it is. And then with the new genetic information, we have now decorated the outside surface to look like the spike protein. And so you're going to be able to train your immune system using this plant virus because it looks like a virus. It looks like SARS-CoV spike protein. What we've also found in the research we've been doing is this virus is so big that it actually gets taken into your cells by the APC presenting cells. And I don't want to go into any more description of that, but basically it will take in this big, huge plant virus. It'll break it apart into pieces. It's going to put it on the surface. So we know it trains both B cells and T cells. So we're getting a subunit vaccine that trains B cells and T cells. So you're saying, well, does this really work? Well, we've been working on it for a while, but we started out like what was important to us at the time was animal vaccines. So our goal has been to basically work on diseases of animals. And that's because Botswana, where we've been working, they have about 2.5 million cattle with only 2 million people. But most of these cattle are owned by smallholder farmers, and that's their whole livelihood. When some of these diseases come through, the smallholder farm suffers badly. And the disease that we went on is something called Lumpy Skin Disease. But before I go into that earlier story, I just want to kind of review what we did. So April, here at KGI, we switched over to COVID-19. We're still progressing on our other vaccines, but this became dominant. And so far we have 20 different vaccine candidates with different parts of the spike protein on the surface of the virus. And again, we're not competing against Pfizer. We're not competing against Moderna. We're working with a place called Botswana Vaccine Institute, and that's in Gaborone, Botswana. And they've been making animal vaccines, but they're partnering with us to make the COVID-19 vaccine for developing countries. So here's, again, Botswana, Botswana Vaccine Institute, and we've been working with the government there and the Agricultural College and the University of Botswana. So let me show you the disease that we've been working on. So it's called Lumpy Skin Disease. And if you could zoom in on that, you would see very lumpy. And it's the bad part about this is these smallholder farmers that have these cattle, you know, they may have only 5 cattle or even less. And it's a primary source of income. This has reduced milk production in the females. The bulls become sterile. There's obviously the hides can't be sold, and then they have death from secondary bacterial infections. So very devastating to smallholder farmers. The important part for me is vaccines, making expensive vaccines is easy to do. Making a cheap vaccine is important. When you're dealing with developing countries, you can't have it like we have with ours. So for instance, the Gardasil human papillomavirus vaccine that we make, if you go to the doctor and you get a dose, each dose is about $150. Of course, we have insurance company. You need 3 doses. So it's with doctor's appointments, you're talking easily over $500. That is something we can't afford in developing countries. So we know that we can get ours for 15 cents a dose using manufacturing facilities that are for good GMP, what they call good manufacturing practice, making sterile vaccines that can be inoculated. But we've been doing this work with cattle, and we did do clinical trials with it, if you will, field trials with it. So we basically, day one, put in our vaccine. Now this vaccine is going to be similar to what I showed you. It's going to have the plant virus that is decorated with a protein that's found on the surface of the lumpy skin disease virus, which is also a very complex virus. Then we test it, pull out some of its blood to see if it's making the antibodies. Remember, those antibodies are specific. We're going to test the antibodies to make sure they're specific against that. So we're going to pull blood at day 14, blood at day 28, and blood at day 42. And at that point, we know that by 42 days, it's making lots of the antibodies that are specific against the disease. So now in our field trials, here's what happens. So if we don't vaccinate it, so here's, we're going to call it day 0. So this cow has not been vaccinated. Then at day 42, you hit it with the live virus, and it starts building antibodies very quickly. So this is the antibody level being made by the animal. You can see it starting to fight and make it. But this is a little too little and a little too late. So basically, the disease takes over. So if you don't vaccinate, this cow loses, and it's going to have the lumpy skin disease. Now let's take a look at the vaccinated cattle. So here at day 1, and again, we're looking at antibody levels. At day 1, we put in the vaccine, and a very fast response. It's making those antibodies that I showed you a picture of. The B cells and the T cells are responding and making it. And then when they've gotten rid of the virus, because as you know, it's a dead virus, it's a plant virus, can't do anything. So it goes back down. But what's important is now we have those memory cells hanging out. They're ready for the disease whenever it comes back. So then at day 42, when we do the live lumpy skin disease virus, boom, it responds very quickly. And it's fast enough and quick enough so that there's no disease in the cattle. And you don't see any symptoms, no indication whatsoever that it even got it as far as it was concerned. The cow is totally comfortable. The only way you can see is by looking at see if these antibodies are being made by the memory cells. So very successful. And I always like to show this slide that shows you the outcome of these. So vaccinated cow, kind of happy. Unvaccinated cow, not very happy. Normally, I also have a picture of a facility that can make literally 4 million plants. Each plant that we use can make between 5 and 10 human vaccines or 1 cow vaccine, 1 or 2 cow vaccines. So very tiny plants. And the facility, this is what the facility looks like. This facility holds, again, 4 million plants. And I'll show you a picture of the plant later. The reason why it's this color is because plants, for those of you who've had botany, plants use only red light and blue light to photosynthesize. So there's no reason in wasting any energy making anything other than the red light and the blue light. So we use LED red, LED blue. The plants grow just fine. They photosynthesize. Everything's good for them. And you just pass carbon dioxide, or just air across them. That's all you need. So that right now, if you're looking at those plants, you see they don't reflect green, which normal plants would reflect green. But since we're not even giving it green light, that's not reflecting any. So they just have the excess colors that we put out there. So here's the plant. It's called Nicotiana benthamiana. It's only 8 inches tall, but this plant again can make enough 5 to 10 vaccines for humans. And so we basically grow these plants up to this size. It takes about 4 weeks to get that size. Then we basically introduce the decorative virus in it. And we have a facility. In this facility, we can basically put this virus in literally thousands of plants at a time. And then 7 days later, we extract the vaccine and purify it and make sure it's sterile. And that's going to be the final fill. The researchers that are in the lab, they're important. In the earlier slide with the guy out in the field trials with the cattle, that was Kelvin Peary. He comes to us via Zimbabwe, and when they started having too many problems, he moved to Botswana, which is where I met him. He did his master's degree there and then his PhD there, and now he's here at KGI as a postdoc working in the lab. So he's responsible for a lot of this work. Karen Paco is a PhD student. She comes to us from Peru, and she's in her last year of making it. And she's also, both of them are very busy, along with Christian. He's a master's student, making these COVID-19 things that are now in animals. And by the end of, well, I guess January 4th, we will know if these antibodies are sufficient enough to stop the virus, and we call it neutralizing the virus. Then we have some undergraduates also in the lab that come in and out. So we do have a lot of great researchers in here. Small scale research, but we really do quite a bit for our small size, and we have a lot going on. The Botswana Vaccine Institute is really excited about making COVID-19 vaccine, and the government is really going to help us also build the facility there. So things are looking pretty good on that. If anyone is interested in helping on this, you can go to the KGI website and the Henry Riggs School of Applied Sciences, and then just direct any donation to Grill Research. I'm not asking for research, but some people are for money, but some people have said, hey, we'd like to do something, and I just wanted to write that down and let you know. So I want to save enough time here to answer questions, because I want to basically have everyone happy that they understand these vaccines and will be as excited to get them as I am. So I'll open it up for any questions, and I haven't been monitoring chat, but it may be someone else who has been monitoring, Jan or Jacob. Maybe you can ask questions or else just unmute and ask a question straight out.
Great. Thank you so much, Dr. Grill. First of all, thank you for your time today. This was so incredibly informative, and your slides were really well presented and clear. So we thank you for putting that presentation together. And also thank you for your work, which is so important to make sure that vaccines are available in developing countries so that we can all have access to the healthcare that we need. What you're doing is so interesting. So thank you to your research assistants as well. We really appreciate it. So we'll open it up to questions. Anyone can unmute, as he said, put it in the chat and we can read it for you. I'm actually going to jump in because I have one myself. There were a difference between the AstraZeneca technique. It was one of the different 6 subcategories compared to Moderna and Pfizer. Does that have any implications in terms of efficacy, in terms of long-term immune response, in terms of anything that they're using different techniques?
So good question. So this, fortunately, this one's been tested before. This not particular vaccine, but this technology. So Ebola, I'm sure you've heard about Ebola. We finally have an effective vaccine against Ebola. It's made using a very similar technology. They took a virus that is symptomless. They put on the Ebola surface proteins onto it. And now anytime there's an outbreak in Africa, they surround that village that has the outbreak and inoculate all the people in the surrounding area. And they call that kind of a circle containment. And we've been able to actually contain and stop the Ebola spread for the first time ever. But it's been out for, I believe, 3 years now, very successful at it. So they're doing the same one. Now, what I don't understand here, and I know that there's been some problems on this, AstraZeneca did some inoculations where they used 2 exact doses, one followed by the other one 2 or 3 weeks later. And the Ebola vaccine only needs to be taken once. And so I don't know why theirs needs to be twice. But then we had found out that they had a accidental low dose followed by a high dose. So the low dose, half dosage, and then 2 weeks to 3 weeks later, they gave them the full dosage. And that's got up to 92% effective, whereas just the 2 high doses did only 62% effective. So they're going to probably do the low dose followed by the high dose. But Johnson & Johnson, who is making a very similar one to the AstraZeneca, they say ours only needs 1. We've done animal tests on it. We've done it on humans already in our phase 2 clinical trials. 1 dose does it. And maybe AstraZeneca only needs 1 dose. I don't know. So I haven't studied theirs enough. But the AstraZeneca is, especially if you get the low dose followed by the high dose, which is what they're going to do, it's a 92% effective, which is pretty good. If you look at any of the other vaccines we normally have, none of them are that effective. So it's actually a very effective vaccine.
So should we be worried or concerned or have an opinion at all about which brand of vaccine we get, or whatever is available to us will be equally effective among the 3 or 4 or 5 options?
Yeah, I'd probably take the first one because, again, the risk of dying with SARS, no one's died from the vaccine. You know, we've had some allergic reactions. Some people think that's the polyethylene glycol in it, but they're not sure why we get allergic reaction. For people who have heavy allergies, they're suggesting either you take EpiPen or something like that, or else just take antihistamines ahead of time. But basically, if you're don't have these high allergy problems, then you're not going to have a problem at all. So that's the main problem we've found with these vaccines so far is the people who are highly allergic. But I would take any of them because, again, the risk of not taking them is much worse. That virus itself is pretty nasty out there. So any vaccine, if I had a choice, I'd probably want to do the messenger RNAs. I'm really excited about those. I think they're just super. Just basically fooling your cell into thinking it's infected, and it's spewing out this spike protein, and it says, hey, this is foreign. It's really doing a great job at fighting back. So I like those. But again, the good part about the AstraZeneca is we've actually had that out in field trials with the Ebola, and it's very effective.
Go ahead, Donna.
Thank you so much. This is such a terrific presentation. What about the mutations, right? We know every year people get the flu shot, and every year they end up with the flu or shingles, same case, because there's a different mutation that they got of the shingles or the flu. What about all these developing mutations now, and how will we need to modify our medical intervention with these mutations?
So it's a very good question. The one thing that we do know is the current way we do the flu vaccine, we're going against the hemagglutinin and the oh geez, drawing a blank here. But anyway, we're going against the wrong proteins that keep slightly changing. And that they're not using that really primarily to get into our cells. It's so easy for them to get into our cells that the mutations keep happening. The one thing we do know about the SARS-CoV-2 is it has a very specific spike protein that binds to the ACE2 receptors in certain different cells. And it's very specific. So far, the data that we're getting on this highly infectious one out there is the spike protein is still binding the same. And so they are expecting the vaccines to be good against the new strains. And that's what we'll keep our fingers crossed that way. The other thing is that at least now, if it does change substantially, we can actually get a new vaccine out pretty quickly because we have this technology now. This is really breakthrough stuff.
Thank you. Thank you.
2 related questions in the chat, Dr. Grill. Are there proof of principle studies of a plant vaccine that can neutralize a virus in humans? And follow up, what is the reason we are not using this type of plant vaccine here in the States? Is there a difference with the more expensive types?
So very good question. I always had this thought that if you make a better mousetrap, that the world would come running. It turns out in the US and in Europe, they don't care about the cost of vaccines. They want to just keep doing it the same way that they're doing. So 15 cents a dose means nothing here. Your insurance pays for it. The insurance company is going to get it paid back. They're not looking to get a 15-cent vaccine out there. In the developing countries, price is an issue. In here, it's not. And basically, I've been told just blunt by the pharmaceutical companies that they're not going to change their system to make it cheaper to make it. That's just not in their, maybe someday it'll change, but right now they have no interest in doing that. So, but again, the other answer is, yes, we do know these vaccines are very capable of doing it. So in my previous iteration before I came into academia, we tried to make inexpensive vaccines, and we found out that, again, the Western world was not into it, and our company had to try and make money. So we switched to making vaccines against non-Hodgkin's lymphoma. And so cancer vaccines, and these are therapeutic vaccines, and I could do a whole presentation on a therapeutic vaccine for cancer, but basically what we would do is we'd go into the lymph node of the person who has the non-Hodgkin's lymphoma, get the cancer and find the protein that's on the surface of the cancer. And it's very specific IgM, it's specific for that cancer, but it's patient specific. So each cancer B-cell lymphoma patient has a different B cell that is cancerous. So we go in, we identify it, then we basically take that and do the same technology that we're doing here, make enough vaccine for that person. So it's a patient-specific vaccine. And we did clinical trials with humans, and we were 80% effective at fighting the cancer with non-Hodgkin's lymphoma.
Great. We have time for a couple more questions. Carol Lee? And then Gus,
You know, and I'm
like...
Canadian, and I'm seeing a lot of discussion in Canada of possibly only doing one dose so that they can vaccinate more people. You touched on that briefly. I wondered if you had more opinions about taking that approach.
Yep. So the point is right now, again, we're losing people pretty fast, and they know they have preliminary information that if you get only one dose of the messenger RNA vaccines, that it's at least 64% effective.
That's better.
Not quite as good, but hey, if you get twice as many people with the same amount, and it's now 64%, you're doing pretty well on that. So they're saying maybe we should just go ahead and give people that, and then follow up months later with another dose. And so just get as many out there to try and get at least 62%, but really follow up with the very critical people with the double dose. But I understand that philosophy. Personally, though, I guess everyone wants to get the 95%.
Thank you. Gus, you had unmuted. Did you have a question?
No, I'll ask later. Larry's my neighbor, so I can ask. It was about, well, actually, I think, Larry, didn't you say you guys are going to file with your vaccine through the European Association?
Yeah, so basically, the developing countries in Africa, they like to do it by the standards of the EMA, which is the FDA equivalent. So it's a European Medical Association. So basically, it's the EMA. They have the same regulatory stringent requirements that the FDA has also, but we would be following the EMAs.
All right, we have a great question in the chat. Can you discuss the concept of herd immunity? What percentage of the population needs to be immune or vaccinated before the community can safely open up?
Yeah, that's a good question. We don't know for sure. Some people have said we need to get up to 90%, but existing data on a lot of other things suggests that 70% can be enough to break this cycle and to finally get rid of it. But that's a good question, but we don't know exactly, but that's the best I could.
Excellent. Well, it's having information like this that's going to help us get to 70% of the population vaccinated. We need to share with people that there's science behind this. It's safe, and it's going to be important for all of us to be part of this next movement of the pandemic.
Yeah,
so
I will be, as soon as I can get there, I'm doing it. When I get my vaccine, I'll be happy.
Yeah. So I know you're monitoring the comments, so appreciative of the depth and breadth, and thank you for your expertise. If we could just give a digital round of applause to Dr. Grill. We'll put this on the church YouTube channel, and so share this with friends and family so that they can have this information. I think it was very valuable. So again, thank you, Dr. Grill.